All right. Well, welcome back, everybody. One of the worst parts about chairing a session is having to kill really good conversation to move on with the program. But we're really excited about the next session. So I'm Andrew Brown from the USA, and I have the great privilege of joining Vasanti Malik from Canada. And this session is on scientific rigor in nutrition research and evidence assessment, strengthening causal inferences through more robust dietary assessment.
And well, it's my great pleasure to introduce our first speaker of the session, Dr. Walter Willett, who we heard yesterday talking about Eat Lancet, but now we're going to hear him talk about approaches to dietary assessment, state of the art. I think a topic that's of great interest to many of us.
Great. Thanks, Vasanti. This is going to be my full semester course in 15 minutes. So bear with me here. Nothing to disclose. To go back in time, in the 1970s, we were looking at data like this from ecological studies around the world showing really major differences in rates of breast cancer, other cancers, cardiovascular disease around the world, strong correlations with dietary factors. And there was a question, is this really causal? And there was clear agreement among people in this area that there was a lot of potential for confounding, and we needed more detailed studies. So that's when I sort of entered my academic career and started working on this big problem. The obvious first thing we needed to do, if we were going to do more detailed studies with better epidemiologic designs, cohort studies, we needed to have ways to measure diet. And those hardly existed. 24-hour recall was almost the only thing that existed and was being used at that time. But that just gives obviously a short period of diet and ignores the day-to-day variability, which was being documented at that time. So I spent in the late 70s a couple of years working on developing a simple food frequency questionnaire, a series of pilot testings, and this is what we came up with: 61-item semi-quantitative food frequency questionnaire is what we called it, and it started administering this to our cohort in the Nurses' Health Study, the first cohort in 1980. We got about 90,000 responses back at that time. But that took a lot of effort to key enter the data manually. It was prone to errors. It was really frustrating to maintain data quality. So we pilot tested a optically scanned questionnaire, and that was really the first time it was used, to my knowledge, in epidemiologic research. And it cut the cost dramatically, cut the error rate dramatically. And so we could double the length of the questionnaire without increasing the cost. This is just a sample of it, but it's about 131 items at that time. And over the years, we've tweaked that to take into account changes in food availability, like low-fat products, et cetera, over time, and changes in dietary choices over time. From the beginning, we realized that there would be a lot of questions about the validity of dietary assessment because people were saying, I can't remember what I ate for lunch yesterday, but I don't care what somebody ate for lunch yesterday. I really want to know what people eat over a longer term, and that's what we were aiming at. So right from the beginning in 1980, we designed a validation study to look at how well we could measure diet, knowing we couldn't measure it perfectly. And this involved 192 people. Were for 4 weeks over a year, we had them keep weighed diet records and then repeated the questionnaire at the end of that period. Because we updated the questionnaire and expanded it, we repeated a validation study in 1986 when we had administered the main questionnaire, the cohort to the main questionnaire again. So to summarize, this is what we saw combining these two validation studies, where we had about half of the people overlapping. They were same people in both validation studies. So very briefly, let's see, I guess my pointer isn't working. Well, yeah. Can you see that? No. We can see in the first column here, that was just if we look at the line for total fat there. Thank you. This was our original questionnaire. the correlation with the diet records was 0.44, and if we adjust for within-person variation in the diet records over time across these two validation studies, it gets up to 0.57. In general, 0.4 is thought to be sort of minimum usable correlation. That's a little bit arbitrary, but as you can see, the 84 questionnaire, which was twice as long, did a little bit better, not twice as good, but somewhat better. And by the time we have 3 food frequency questionnaires, average those and look at the multiple diet records over the 6-year span, we get a pretty high correlation, actually 0.83. We're really not missing very much, it looks like, in assessing total fat over the 6-year period. And it really is that multi-year period that is of importance to us. It's not one day, but it's not even 1 year when we think about the origins of cardiovascular disease or cancer. Subsequently, thanks to the 2008 recession, we got the funding to do a validation study we'd really like to do. It probably cost about $15 million, largely due to the doubly labeled water measurements that we had in there. In this validation study, this is women, we had about 700 people, and we had a parallel study in men of about 700 people as well. And we had, again, the 2 food frequency questionnaires, one at the beginning, one at the end. We had 2 7-day diet records where participants weighed and measured everything they ate, and then 4 24-hour recalls using the online ASA 24 method. And then doubly labeled water on some people, a subsample, repeated measures of that. And we had 4 24-hour recalls, 24-hour urine samples, and 2 fasting blood samples. So we could really look at a lot of biomarkers. I won't mention all of them, but with repeated measures so we could assess within-person variability in everything we measure. And I won't go into the timing, but that was all very carefully done. So we didn't measure a biomarker at the same time we had a short-term measurement of intake, because you get falsely high correlations if you do that. So just very briefly, looking at what we looked at over time in the other validation studies, the correlations with our questionnaire and the weight diet records was about 0.63. And that's really what we saw back in 1980 and 1986 in our validation studies then, which was good because people had said, well, the food supply is changing, people are eating out, they don't know what they ate. It looks like our tweaks along the time had been able to keep up with what was going on. Interestingly, if you use the 24-hour recalls and de-attenuated for the within-person variability, we came up with almost the same correlation, which says you can do validation studies that cost a lot less, actually, which is important. The confidence interval is wider because we only had 4 days versus 14 days with the weight diet records. If we included supplements, the correlations were a little bit higher there, as you might expect. One of the things we could do, where we had the 3 major ways that diet has been measured, 24-hour recalls, food frequency questionnaires, or semi-quantitative food frequency questionnaires, and weight diet records, this allowed us to look at the relative validity of these different methods, which had really not been compared head-to-head before. And we could do that because we could compare each of their correlations with the same biomarker, which gives an independent measure. It's got its own errors, as I'll talk about, but it allowed us to look at the relative validity of the different methods. That generated a lot of data, and I won't go through all of that for sure. Beta carotene is a pretty good example, though, of in between the worst and the best, but basically, this is the correlation, de-attenuated correlation with a biomarker. The gold column here is the 2 weeks of weight diet records. And that consistently, if you really have unlimited resources and unlimited capacity of participants to make the effort to complete this, that is really the gold standard. Interestingly, this is 1 week of weight diet record, and in general, it seems to be about as good as our food frequency questionnaire, or the average of 2 food frequency questionnaires. That's the summary of everything we looked at comes out to be about that. And the diet record costs about 1,000 times more per person than the food frequency questionnaire, because we can scan it and process it electronically. So it's a pretty good deal, it looks like, there in terms of having a method that will work in large studies. That analysis was for nutrients. We've done validation studies on the basis of foods as well. And on average, the correlation with foods and the diet records is about 0.7, but quite a bit of variation here. The vertical column is a correlation for different foods with foods, same food assessed by the diet record. So quite a bit more variation. Basically, things that people eat, consume often, a lot of beverages down here, and episodically consume foods have lower validity correlations, but it's probably because the diet records are less than ideal for those episodically consumed foods. We've also looked at the validation for dietary patterns. See, these are different dietary patterns that are familiar to you. And there the correlations tend to be a little higher, about 0.75 for the different dietary patterns, and all seem to have pretty same validity, not too surprisingly. Now, how can it be that something that seems so primitive as a food frequency, semi-quantitative food frequency questionnaire, how can it be work that well compared to things that cost a lot more time and monetary cost as well? And the reason is that we're basically powering this by the human brain, and we're using that to extract a lot of information that's been stored in ways that you can't capture in other methods, including like average intake over a past year. You're not going to get that from any of these wearables or anything like that people are using. There's someone at Harvard estimated there's 86 billion neurons and 100 trillion connections, and not to use that just seems like a waste of available information, and that's what we're pulling out with our questionnaires. But the time is really important, and longitudinal changes in diets are really important to assess. Just for example, this is looking at use of different oils in the health professionals follow-up study over just a 20-year period, and we're looking over a 40 or 50-year period now. But corn oils, soybean oil consumed in our cohorts gone way down over time. Olive oil, canola oil gone way up over time. And even more complicated, the processing has changed radically over time. We have a dynamic database, food composition database, that we maintain for our questionnaires. So every 4 years, we've gone and gotten the products off the shelves and analyzed them for fatty acids, gone to Burger King, McDonald's, and pulled out their French fries, even paid for them, in fact. And we found in 1991, the trans fat was 30% of the fat in their French fries, and you can see that's gone stepwise down over time. By 2011, it was basically gone. So if you don't take into account both what people are eating, but also the composition of foods, you can basically lose a lot of information. So one of the analysis that we did was looking at type of fat in relation to total mortality in our cohort. And our default method is to use cumulative update, where at any one point in time, say we're looking at incidence of death from 1990 to 1992, we use the average of all the previous questionnaires to get the best, most complete information over the previous years. But it's always prospective. So always we're looking at diet before the occurrence of the outcome. And this is what we published. And I saw with here about 30,000 deaths over 30 years. Trans fat by far the worst. This is different fats were compared to carbohydrate intake isocalorically. Saturated fat, modest increase, but highly significant with these numbers. Everything's highly significant. MUFA inverse, very strong inverse relationship with N6 PUFA. But this is what we saw if we only use the baseline. And there are basically missaturated fat, mistrans fat, MUFA in the wrong direction, weak inverse relation with PUFA there. So you miss almost everything if we only had the baseline information. And unfortunately, most of the cohorts around the world only have the baseline information. So they're losing a lot of information. Another thing we can do with repeated measurements is look at latency with a lot of statistical power. And not surprising, for some cancers, it takes quite a while for the association to show up. This is, for example, you don't see much if you're looking just at baseline with folic acid and colorectal cancer. But with a latency of about 12 to 16 years, then we start to see a quite clear inverse relationship that has continued over time. Also, we can look even more in retrospect at adolescent intake, and we've done some validation studies where while women were relatively young in the Nurses' Health Study too, we asked them to fill out a questionnaire specifically designed about foods that were available while they were in high school. And interestingly, for breast cancer, those are the strongest associations that we see, the high school diet, not their diet during midlife. And this is, for example, red meat intake. Just in the last few months, we've done some analyses looking at how do we enhance our dietary assessment. And there's several different strategies that with any measurement, you first try to get your instrument optimized the best you can. Second, you can use repeated measurements and take the average of the repeated measurements. And third, you can calibrate your measurement to the best gold standard. And so that's what we've done here. This is looking at red meat and coronary heart disease. I showed the data for diabetes briefly yesterday, but if we only had baseline looking at processed red meat here and unprocessed red meat, baseline only, it's a modest increase in risk here, and you would say this is really pretty null, not significant for unprocessed red meat. But if we calibrate the baseline assessment, just only baseline, there's about a doubling of the excess risk at relative risk -1. But we don't gain statistical significance by calibration. And we start to see a little bit of increase here with unprocessed red meat. But if we use repeated measurements, there we about double, again, the excess, the effect size, the relative risk -1 compared to the baseline. But we've got a lot more information, so it becomes more statistically significant. And for unprocessed red meat, that additional information does show there's an association here that's actually pretty highly significant. And then if you do both, we have repeated measurements and then calibrate them. And we only have had the method available for the last year or so for doing that. Then it looks like there's about a 3-fold increase in the effect size estimate here and about a 5-fold increase there for the unprocessed red meat. And it's a little bit sobering because this is what's used in most of the literature. And if people do a meta-analysis, that's what they're mostly getting is studies that have only had baseline information. But this is partly emanated out from some of our work in Eat Lancet, realizing that a lot of the literature, if you just look at meta-analyses, there's pretty severe underestimation. Okay, wind up really quickly. There's some novel methods here. I won't go into, hopefully other people will talk about it, but I want to emphasize that they really do need to be looked at critically for validity, burden, cost, compared with other methods. And the same with biomarkers as well. They're attractive, but they all have their limitations, including a lot of homeostasis, feedback, and basically affects the metabolism of those nutrients. And one good method is using to compare them again, the method of triads. And here, this is sort of the poster child of metabolomics, is proline, betaine is an estimate of citrus food intake, but you can see in the method of triads, the food frequency questionnaire does quite a bit better, about the same as the diet records than the method for the metabolomics measurement. Our best example here of an integrated time measurement, toenail selenium, because that's integrated over years. So just to summarize, the optimal assessment will depend on the intended use, specific aspect of diet, population, participant burden, cost, et cetera. There's no perfect method for all the things we might want to do, and those all need to be considered. But current diet assessments actually work pretty well to detect important associations. Repeated measurements are important in long-term studies. We need to keep an eye on childhood, diet, and adolescence. Biomarkers can be really helpful, but not all the time. It needs to be looked at nutrient by nutrient or variable by variable. And in the bottom line, I think we can say that cohort studies combined with short-term feeding studies with risk factors as endpoints can provide strong evidence for translation to recommendations and policy. Thank you.
And so now I'd like to introduce our third speaker, Clemens Wittenbecker from Sweden, who will be speaking about metabolic adaptation to dietary macronutrient shifts and impacts on objective assessment and health implications. Welcome, Clemens.
Hey, yeah, thank you very much for having me here. Thank you to the organizers for this great conference. Yeah, I will shift the focus a little bit more towards like dietary influences on diseases, and I have no conflicts of interest to declare, but I'm also interested in the effect of fat, dietary fat on health. And if we like the view of this has shifted from total fat as a factor that increases LDL cholesterol and thereby influences cardiometabolic risk to a more nuanced understanding where different fats play a different role on different aspects of both lipoprotein metabolism, but also non-lipoprotein related influences of lipids on cardiometabolic risk. And I think it's quite plausible to assume that metabolomics, or especially lipidomics, is quite like a promising tool to basically capture this more comprehensive effect on cardiometabolic risk of dietary fat more completely or more comprehensively. And with Walter's introduction, I feel that it's also important to justify why do we need these molecular markers, and beyond like insight into mechanisms, I think it's also a strategy to just corroborate causal claims of dietary influences on disease risk, because if we break that diet disease association for many of the complex exposures, we cannot conduct endpoint trials. If we break it into smaller pieces and just demonstrate the effect of diet into robust metabolic adaptation outreach in RCTs, and then use these markers for metabolic adaptations to examine their long-term disease risk associations in prospective cohorts, then we can generate pretty solid evidence for causal effects. But it is also important to generate better biomarkers for translation into diet counseling and clinical practice, because we want to have markers that actually respond well to diet and at the same time where the changes predict changes in subsequent disease risk, because we need it to inform prevention. And if there are subgroups of the population that benefit the most of specific dietary interventions, it's also useful to have objective tools to identify these subgroups that respond with a risk reduction to a dietary intervention and distinguish them from those which might be require different prevention measures. And yeah, I have talked about our study on dietary fat quality already on the last DNSG conference, so I will go quickly through it. But since we have these discussions around seed oil, I feel I still mention it. We have basically conducted the study with a very thorough combination of a dietary intervention trial here, the DIVA study, which is an intervention trial with where every participant receives the same amount of total dietary fat, but in the control group, about half of that comes from dairy saturated fats, and in the intervention group, most of the saturated fat is substituted with plant-based unsaturated fat. These dietary intervention is pertained over 16 weeks. We have lipidomics data from before and after the intervention, and based on the changes in 45 lipid metabolites that are significantly changed between the high saturated fat and the unsaturated fat group, we develop a multilipid score, and then we reconstruct this multilipid score in several prospective cohorts, including the nurses' health studies. But here I show data from the EPIC Potsdam cohort study, where we associate the multilipid score with CVD and type 2 diabetes incidence, standardized on the effects that the dietary intervention in the DVAS trial had on the change in these multilipid scores, and compare that to the change in non-HDL cholesterol. So as expected, the diet intervention in the DVAS trial also reduced LDL cholesterol, but it appears here that if we extrapolate risk with this more comprehensive lipidomics outreach, we basically extrapolate a risk reduction of about 30% of CVD risk and over 20% of diabetes risk reduction. And that's a much stronger risk reduction than we would extrapolate with the effect on non-HDL cholesterol. And also interestingly, if we include both the multilipid score and non-HDL cholesterol in the same already confounder adjusted model, it explains the association of non-HDL cholesterol, but not the association of the multilipid score. And then interestingly, we also went into the Predimed trial and conducted interaction analysis between that multilipid score and the dietary intervention in Predimed, which also reduced the incidence of type 2 diabetes. And we see a significant interaction and follow that up in stratified analysis. And we really see that this diabetes protective effect of the Mediterranean diet intervention is particularly strong in those participants who had an adverse multilipid score before the dietary intervention, whereas those with already were more on the beneficial side of the spectrum of that multilipid score did not benefit or respond with diabetes incidence reduction in the same way. And that is not explained by a large number in the incident cases. So it's not just a high risk identification within that, but really appears to highlight those which are more responsive to the dietary change. This has been published. And now we follow the same strategy on another quite publicly discussed topic, the replacement of carbohydrates with fats, so low carbohydrate diets. And again, we looked for a dietary intervention trial here, the Framingham Food Study, where we have 3 groups with continuous basically exchange of carbohydrates with fats. So the highest carbohydrate group has 60% carbs and 20% fats, and the low carb group has 20% carbs and 60% fat, and one in the middle. And then we align the metabolomics data here with 4 cohorts, including the 2 nurses' health studies, but also the Jackson Heart studies with African Americans and the Mexico City study, and look into consistency of the diet associations in substitution models, but also relate the metaboloids to mortality and type 2 diabetes risk. And this is just to show you that we work in the cohorts in a little bit different range of total carbohydrate intake in terms of percent energy. We do cover the 40 and 60% of carbohydrate as a source of total energy, but almost no one has an intake of only 20% of total energy from carbohydrates. And what we see if we look into the metabolites that were first affected by the FS2 intervention trial, so the low carbohydrate dietary intervention, and then only look at those metabolites in the prospective cohort studies and use dietary substitution model, where we actually model the replacement of carbohydrates with fat. We see actually a very impressive consistency. So from the 87 metabolites that we had across the trial and the cohort. So the trial is here. Do you see the pointer? Or well, the inner ring here is the thank you. So the inner ring here is the trial effects. This is the meta-analysis of the 4 cohort studies. And we see basically a large match of colors, which is translates basically in 98% of the metabolites that are directionally consistent between the intervention effects and the meta-analysis of substitution models in the cohorts. And over 80% are also statistically significant. And we focus on these basically consistent metabolites and subsequent analysis. But the cohorts also give us more flexibility in modeling. So we can then go, because we have the variance in that large population study, we can model replacement or effects of replacing carbohydrates with only saturated or polyunsaturated or monounsaturated fats, but still focusing only on the metabolites that we before that validated in the or discovered in the trial. And we see quite interesting differences. So let me just as an example focus here on the triglycerides, where we have a reduction of triglycerides by substitution of carbohydrates with fat in general. But if we look in, for example, saturated fat specifically, it actually only reduces the triglycerides with a high amount of unsaturated fat. And the ones with more saturated fat tend to increase, and the opposite picture of carbohydrates with unsaturated fats. And we also then look more into detail into the differences between plant and animal-based monounsaturated fat. But interestingly, if we overlay then the diabetes associations of these metabolites with the low-carb diet intervention effects, we do see that in general, low-carb diet tends to decrease the metabolites that are associated with higher diabetes risk and decrease those which are associated with lower diabetes risk, but it's not completely uniform. So there are some exceptions, some red in the right half and some blue in the left half of the plot. And if we then look into the fat type specific effects, you can see that the unsaturated fats are like seem to be more effective in reducing those metabolites that are most strongly associated with type 2 diabetes risk. But yeah, that might this like mix of effects is much clearer if you look into the association with mortality, where you don't have like much consistency. So among those low carb diet affected metabolites, there are 18 associated with mortality. And you see some of the high mortality associated metabolites increase with low carb diet, some decrease, and likewise the low mortality associated metabolites, some increase and some decrease with low carb diet in general, if we look at total fat. But this becomes much clearer if we look into the substitution of carbohydrates with specific fat types, because here we really see that saturated fat seems to increase detrimental metabolites and decrease those with associated with lower mortality, whereas unsaturated fats decrease high mortality and increase low mortality associated metabolites. And then we can again summarize that into signatures and look into the association of metabolite signatures from these trial validated metabolites with for total animal-based monounsaturated, plant-based monounsaturated, and polyunsaturated fat. And for diabetes, we don't see much, but a strong risk or a risk reduction through in relation to the metabolite signature of plant-based monounsaturated fat. And for mortality, we do see that total fat for carbohydrate replacement seems to be detrimental, mainly driven by saturated fat, whereas the substitution of carbohydrates with plant-based unsaturated fats is beneficial in terms of mortality. So to sum up, I think echo your conclusions to some extent. I think these metabolomics and lipidomics profiling studies can really give us insights into biological processes, especially if we look into the combination of intervention trials and cohort studies, understanding these metabolic adaptations and also using the flexibility of cohorts to model more specific dietary intakes can help us to also inform dietary risk prevention strategies more specifically. And I also think that they can be a resource for prediction and monitoring biomarkers. Yeah, I want to thank all the people who were involved in these studies and especially you for your attention. Thank you.
All right, thank you very much. So next up, we have Sangeeta Shyam from Spain, who will be talking about the impact of investigator bias in nutrition research.
Thank you, organizers. Thank you for giving us this opportunity to present this really interesting topic here. And before I proceed, I would like to give you my disclosures. I currently hold a position which is financially supported by the Carlos III Health Research Institute in Spain and is also co-funded by the European Union. And prior to moving to Spain, I have received industry grants. And I'm also a member of the Federation of European Nutrition Societies working group, which is developing the Consort Nutrition Extension. And I lead the activator meta research for Asia. So all of this could have somehow influenced some of the ideas that I'm going to be presenting here. And the most important disclaimer is this is coming out of an opinion piece that Professor Jordi Salas and I wrote. So it's an opinion piece. So all of us have the ability to hold opinions. So you are free to disagree. And for the sake of this conference, I'm also bringing in opinion from other active researchers in the field. So we broaden it out a little bit for you. So basically, the presentation outline would be as this. So I would like to tell you about what investigator bias is from our perspective and why should we even care. And then I'm also going to talk to you about some examples as to how investigator bias can affect nutrition research and some brainstorming on the ways to move forward. Okay, what is investigator bias? This is not something new. We've been thinking about this for decades, but we've not been doing much about it. So in 2003, Ernst and Cantor, they would say that bias resulting from a conflict of interest that arise from passionate beliefs was something that was called investigator bias. And they linked it to positive findings, interestingly. And Jordi and I felt it was not necessarily so. And we chose to define it as acts of omission or commission arising from ignorance, hubris, or excessive attachment to beliefs. For those of you who do not have beliefs, hubris, or ignorance, feel free to leave this presentation because this is definitely not for you. These are only the three main factors that we think affect, but there could be a lot of different factors. So once again, we are not experts in investigator bias, so please excuse us for that. And I would also like to place the presentation in context that we are not just referring to research misconduct or unethical publication practices. Okay, so why care about this? Why do two people who are sitting on a data mine and having a lot of things to do, writing an opinion piece which will not be even considered as part of their CVs? Because we actually care. Jordi and I usually happen to be the last people moving out of the unit, and we spend this time talking about different kinds of things that we could be better at. And that's how this whole thing started. And I think the discussion started on the plane ride back from DNSG Sweden back to Reus. And here's somebody who's way younger than the both of us, who also raises concerns about investigator bias. And he says, a certain amount of it is all right, but when it crosses the limit, can you hear me, Professor Michelin? Am I not clear? Are you able to hear me? Yeah? Yeah, okay. So when the bias becomes a bit too much, then it distorts interpretation, it raises correction, and ultimately hinders discovery. So yeah, I was working in an international medical university. So there, the nutritionists are the poor cousins of the doctors and even the pharmacists. And so most of the times you would hear people say this to you. So today, which spinning wheel are you spinning? For your exposure, for your population, and for your outcome. And so it was funny for most of them. And this is a graphic that's coming from Darren Daly, and he has given us the permission to use this in this presentation. And more importantly, so even in academic scholarly literature, you have people who talk about disclosures in nutrition research, investigator bias, etc. So if it remained within our coffee corridors or even within our scholarly publications, it's not so much of a problem. It's an irritation. It's not so much of a problem. But unfortunately, it doesn't end here. So what happens is when this plays out in the public domain, there is weakened trust, public trust in nutrition. And we are also creating poor evidence for nutrition, and it leads to poor population health and nutrition, which is why we definitely should care. And so it was really fantastic that the Federation of European Nutrition Societies and the American Society of Nutrition made some efforts to make this better. The Federation of European Nutrition Societies came up with certain working groups which worked on improving methodologies, a working group which is working on improving scientific communication, both in scholarly publications and also in terms of media reports. So the media reports one is already ready. So if you're interested, you could go to their website and look at their checklist for how to improve reporting. Using the media outreach that you would do. And the American Society of Nutrition identified several domains which were linked to public trust in nutrition. These include accountability, conflict of interest declaration, scientific rigor, reproducibility, equity, and transparency. And it's not even triangulation. You can see how investigator factors can affect all of this. So definitely investigator bias affects public trust in nutrition, and we all have to strive for that. So therefore, conceptual understanding of what investigator bias is very important. At this point, we do not have conceptual frameworks to understand this. We need to know what is the prevalence of this kind of bias in our literature, because we are talking about AI, we are talking about machine learning, and it learns from us, unfortunately. We need to understand the drivers and the perpetuators of this bias, and we could probably propose some safeguards for this as well. So just to give us some examples, all of us definitely know it's just a small period for you to sit back, relax, and self-reflect and think of those papers which annoyed you. So these are people who have shared their thoughts as well. And you will look at how people view investigator bias as affecting various aspects of nutrition research. The most frequently identified investigator bias comes from the statistical tests. We shake the tree. As my friend Hector says, sometimes, we don't want to shake the tree till the P is less than 0.05. So data analysis is an important point. Another colleague who does not work in Spain says that reviewers can be a really important source of investigator bias, and they kind of are gatekeepers of science, and this is a big problem as well. And there's one about UPF, which is very interesting. Media hype and funders being interested in actually funding certain buzzwords is also a problem, because people go into that without checking definitions and assumptions as well. And another person, very senior person, says the places where she can actually identify investigator bias is in the introduction and in the discussion, because you only cite people who support your findings. Yeah. And here are other examples. These are not, by no way are they exhaustive, just for you to have a look. And so when Jody and I looked at stages in the research where investigator bias could affect, it was everywhere, right from the research questions to formulating the research design, to how you analyze the data, how you interpret it, or how you review other people's work or provide feedback. All of this is affected. So let's look at a couple of examples. Asking question no one wants the answers for. doing redundant research reviews, right? And sometimes asking questions where you have millions of funding to find an answer which is not going to improve population health. I'm not going to give examples, and this is a deliberate decision because it's harder when we provide actual examples or cite papers, because it's more easier for us to understand the concept and accept it if we didn't do that. So the next one is adopting inappropriate methods. There was very recently a very, very famous trial, which most people thought, probably the investigators didn't claim, was supposed to be answering about precision nutrition, but it ended up creating so much of discussion in the social media because they had bundled a lot of things together. When people meet healthcare professionals more frequently, yeah, and when they log their diets, their physical activity, they are likely to do better. So was precision nutrition actually the one that caused the treatment effect? So these kind of things are what you would think leads to inappropriate adoption of methods. And then manipulating the analysis, I think we are really very aware of this, so I'm not going into this. So some of the reporting guidelines now even they require you to justify your modeling choices, which I think is a very great idea. So interpreting inappropriately, we've looked at this already, and sometimes we like to exaggerate small effects because we have small effects in nutrition, right? And then finally, providing poor evaluation and feedback. Affirming work that aligns with what you think it should be and rejecting alternative views is a real big problem in nutrition. So how do we move forward? Nothing great, just reflect, accept, and probably we should look into ourselves, ponder on what drives and perpetuates this, consider what safeguards we already have, and consider what safeguards we could have. So when I spoke to you about this really controversial paper that came up talking about disclosures in nutrition research, the European Journal of Clinical Nutrition, the editorial board was definitely upset, and they came up with this editorial where they say there is a clear need for self-reflection by the scientist prior to embarking on industry-based research. And here's where I want you all to pause, and they don't stop at that. Have a look, and they say for the scientist who seeks to prove their favored hypothesis as well. So it's not only those who are sponsored by the industry, but others who have their pet favored hypotheses as well. So here's another professor who says we need to first realize that we are doing this. So reflection is very important, which is why probably Jody and I spent those few months writing this paper just to create that, sensitize and create awareness. To understand what perpetuates and drives this bias, there's a lot of different things. So this is coming from a professor in Asia. You see there's a lot of different things here. And when we broke it down in our publication, we saw a lot of different things. It was coming from systemic and cultural aspects, from individual aspects, and procedural challenges. We do not have a checklist kind of like, I don't think this is a problem this morning. We even saw that it is hard to define what's remission. So investigator bias is even more harder for us to understand what it is. Biases in the funders, reviewers, and researchers ourselves perpetuates this some more power hierarchies. So if you're working in an environment where you're scared to stand up and say, this is investigator bias, you're going to perpetuate that as well. Our ranking matrices and all of this perpetuates the bias. Potential preventive measures, these are just a couple of ideas. Some of them already exist, some of them have been repurposed, and some of them probably are just ideas that we are putting out, which may or may not be useful. So when it comes to formulating research design, I did notice that we are increasingly looking at participatory approaches, using patients' participation into coming up with questions, and also actually involving the stakeholders. Also, having boards that vet more than the scientific validity for also things like potential biases that are there in the protocol would be useful. Second thing about formulating study design, we tend to work with the same people over and over again, people who are very similar to us, and that is very natural, but sometimes having a diverse team just talking to somebody who's a little bit diverse than who you are is very, very useful as well. Analyzing data, here's something that we could definitely do, and most of us are already doing, having a pre-specified statistical analysis plan and sticking to it. And then interpreting findings in the discussion, managing to say that we have had a pre-specified plan or acknowledging that we didn't have it and this is pretty exploratory in nature. And also while providing feedback, we usually ask the authors to, list down all their conflicts of interest, but we never ask the reviewers to do so. So that's something we can think about. So some ways forward, okay. Some ways forward that we could do, probably most of you are mentors, most of us are mentors, encourage reflection. And if we are part of professional societies, look at ways we can, sensitize all the stakeholders. And if you are in an institutional level where you have the ability to influence, probably build tolerance to alternative views and embed diversity in your systems. Here are my last words. Our last words, if I could say so. So being prescriptive and asking people to just disclose their conflict of interest has never been a solution to anything. So probably our best bet yet is to train the younger generation of scientists to be self-aware. And this is what we thought we are going to do by this presentation, to move the thinking from that end to this end, where people think we do a little less shit than we actually do. So with that, I would like to thank the organizers and Professor Jody Salas again for this opportunity, to all these people who gave their time to send in their views, and all those who are financially supporting me at this point. Thank you very much, everyone.
Thank you very much, Dr. Shyam.